Hello, class, and welcome back to the second part of Lecture 3 in Human Development Theory and Practice, where we will be taking a look at DNA and genetic and brain development in this second part of this lecture. So next we'll delve into that captivating realm of genetic foundations that underpin prenatal development. So our journey takes us through that intricate world of DNA and genes and chromosomes, that symphony of genetic information that guides the formation of life. So let's take a look at DNA and genes and chromosomes and have a better sense of what these are. First off, DNA. What is DNA? DNA is, in essence, the blueprint of life. It is deoxyribonucleic acid, and that's why we call it DNA, and that stands as the fundamental code of life. And it's a remarkable molecule composed of long chains of nucleotides, each containing sugar, a phosphate group, and one of 4 nitrogenous bases, and it forms into a double helix structure. Then we have genes. And what are genes? Well, genes are essentially units of heredity. Genes are specific segments of DNA that carry the instructions for creating proteins, the workhorses of the body, and these proteins play pivotal roles in nearly all biological processes, from cellular structure to enzyme functions. And genes are basically like chapters in a book, with each chapter providing information for a specific protein. And humans possess about 20 to 25,000 genes, and each gene governs a particular trait or characteristic. Now, let's take a little bit of time and explore how these genes work in creating who we are, and particularly in the realm of prenatal development. So, with this, there are dominant and recessive traits. And we need to kind of understand the essence of dominance and recessiveness, and we've mentioned a little bit in our previous portion of this lecture. But essentially, genetic traits are determined by specific versions of genes known as alleles. And alleles can be dominant or recessive, and their interactions dictate the expression of traits. Dominant alleles mask the effect of recessive alleles when present together in an individual's genetic makeup. Now, there's something known as Mendel's law, and Gregor Mendel, who was kind of the father of modern genetics, uncovered the principle of inheritance through an experiment with pea plants. And it's something we can apply to humans. Now, what he discovered was that some traits are dominant, expressing themselves even in the presence of a recessive allele. For instance, if an individual inherits a dominant allele for brown hair from one parent and a recessive allele for blonde hair from another, the dominant brown hair trait will be the one that's observed. But there are recessive traits, and they can occasionally express themselves. And recessive traits are only expressed when an individual inherits 2 copies of this recessive allele, one from each parent. And this results in the absence of the dominant allele's influence. An example is the inheritance of blue eyes, where both parents contribute to recessive allele for blue eyes, leading to an expression of in the offspring. Now, there's something else that we need to kind of be mindful of in this dominant recessive interaction, and that is polygenic inheritance. Now, while dominant recessive traits involve the interaction of a single gene pair, many of our traits are influenced by multiple genes, and that's why we call this polygenic inheritance. That refers to the additive effect of several genes on a single trait, contributing to the continuum of variation that we observe in various characteristics of people, like their height, their skin color, and even their intelligence. Then there's the complexity of polygenic traits, because everything impacts on everything else. And so polygenic traits result from the combination of actions of multiple genes, each contributing a small incremental effect. As more genes contribute, the trait's expression forms kind of a bell-shaped curve reflecting the diverse range of phenotypic outcomes that we often see within a population. So, some examples of polygenic traits, things like height. Height is influenced by an interaction of numerous genes, each contributing its own small effect. And taller people tend to have taller offspring due to the cumulative impact of these genes. Something else would be skin color. Skin color is determined by the interaction of several genes that influence the production of melanin, the pigment responsible for skin color. The results are obviously a wide spectrum of skin tones. And intelligence, too, is something that can be impacted by chromosomes. So intelligence is another example of complex traits that are influenced by a multitude of genes, each contributing to the cognitive abilities of the individuals. So this polygenic inheritance really kind of explains the range of intelligences that we see within a population. Now, what is a chromosome? You can kind of think of a chromosome as the genetic library, where genes are organized into structures, and we call these gene structures chromosomes. So humans have 46 chromosomes with 23 inherited from each parent. And these chromosomes are divided into pairs, with one chromosome of each pair coming from the mother and the other coming from the father. So the chromosomes really kind of serve as this repository of genetic information, ensuring the orderly storage and inheritance of the genes. So it gives some structure so that the body can kind of find the information that it needs. Now, chromosomes are further organized into specific regions and bands. Karyotyping, which is a technique used to visualize and analyze chromosomes, provides a snapshot of an individual's chromosomal makeup. And so abnormalities in chromosome number or structure can lead to genetic disorders such as Down syndrome, which is caused by an extra chromosome of the extra copy of the chromosome 21. Then there is genetic diversity and variability. And here, the variability in human traits arises from the diverse combinations of genes inherited from the parents. And this genetic diversity is essential for both adaptation and evolution. And with the Human Genome Project that was completed in 2003, it mapped the entire human genome, shedding light on the mass amount of genetic variations and providing insights into disease susceptibility and drug responses. So there's a lot of information that we now have about the human genome. Now, let's go on and explore a little bit of what happens in the early embryonic development, because that's really fascinating as well. So, when we have the fertilization process, we have the gametogenesis, which is the formation of the eggs and sperm. We've already talked about that. And then at the moment of fertilization, we have the formation of the zygote. And with the formation of the zygote, we have that fusion of the genetic material from both parents. And it contains then a complete set of 46 chromosomes, 23 from the mother and 23 from the father. This genetic combination defines the individual's unique traits, characteristics, and potentials. Then we have blastocyst formation and implantation. And we've talked about this a little bit as well, but I want to kind of delve into it a little bit more. So first we have that rapid cell division. So after fertilization, the zygote embarks on a journey of rapid cell division. And this cluster of dividing cells, known as a morula, eventually forms the blastocyst, that hollow ball of cells. And this early stage of embryonic development marks that transition from a single cell to a complex multicellular organism. Then we have that process known as implantation. So the blastocyst travels through the fallopian tubes to the uterus, where it undergoes a critical process that we call implantation. And during implantation, the blastocyst attaches itself to the uterine lining, establishing a connection that enables the exchange of nutrients and waste products between the developing embryo and the mother's bloodstream. And there's the formation of the amniotic cavity and the embryonic disc. So as implantation progresses, the blastocyst undergoes further transformations, and a fluid-filled cavity known as the amniotic cavity forms within the blastocyst. And this cavity encloses the embryonic disc, a small structure consisting of two primary germ layers, the hippoblast and the epiblast. And the hippoblast gives rise to extra amniotic tissues, while the epiblast will continue to the formation of the embryo itself. So you have this kind of interesting combination of things going on there. Then we have the development of the outer layer of the embryonic disc, known as the epiblast, that gives rise to a region that contributes to the formation of the placenta. And the placenta plays a pivotal role then in providing oxygen, nutrients, and waste disposal for the developing embryo, facilitating a nurturing environment within the uterus. And so we have this fascinating process that goes on here that it really is something that's really wondrous. Now, we've talked a little bit about how things work normally. What I want to do now is just talk a little bit more about the brain formation process, because it starts off with a neural tube. So we begin with the blueprint for the nervous system. And the nervous system really is a complex network of cells that enables communication and coordination within the body. And the foundation of this intricate system is laid during the early stages of prenatal development through the formation of the neural tube. Now, how does this take place? Neural tube formation is a pivotal event that occurs in the embryonic stage around the third week after fertilization, and begins with the transformation of the ectoderm, the outermost germ layer of the embryo, into the neural plate, which is just a flat, simple plate. And this flat structure elongates and then slowly begins to form inward, and it eventually transforms itself from this, think of a flat piece of paper that folds at both ends, and eventually you have a tube instead of a flat piece of paper. That's basically what you have here, is over a short period of time, that neural plate encloses in and creates a neural tube. And that neural tube then is the precursor to the brain and spinal cord. So it's obvious that the formation of that neural plate is extremely important, as well as the folding process. Extremely important. And anything that disrupts either of those will have profoundly devastating consequences on the embryo that forms later on. So there's something that we can think of in terms of special migration. So in addition to the formation of the neural tube, another remarkable process unfold simultaneously with this, and that is neural crest cell migration. And that is these cells derived from the neural tube start a journey to various regions of the embryo. And as they migrate, these neural crest cells contribute to the formation of various structures, including facial bones and cartilage and some components of the peripheral nervous system. So there's some movement around of these cells that form various parts and various destinations. These cells can be responsible for the pigmentation in the skin or the sensory neurons, or supporting cells for teeth. And then there's also the formation of the primary brain vesicles that go on. But as I mentioned, I want to talk a little bit about what happens when neural tube development goes awry. Now, in that early stage of prenatal development, that neural tube is a fundamental structure that gives rise, as I mentioned, to the brain and spinal cord. And the process of neural tube closure is a very tightly orchestrated symphony, and it involves very precise folding and fusion of tissue. However, when this process encounters disruption, it can lead to neural tube defects. And you can see some of the pictures on the slide about this. And it can give rise to something known as anencephaly. And one tragic example of this disrupted neural tube development is anencephaly, which is a severe and often fatal neural tube disorder. And in cases of anencephaly, the neural tube fails to close at the cranial end, resulting in the incomplete development of the brain and skull. And as a consequence, a major portion of the brain, including the cerebral hemispheres, fail to form properly. And so there's obviously going to be some devastating developmental consequences of this. So the prognosis and outcomes for anencephaly typically are that there are profound cognitive neurological deficits, and oftentimes the infants are born have little or no brain tissue covering the exposed cranial area, and often they'll have things like what is called an open skull, which basically is what it says. You can see the top, you can see their brain at the top of the head. And so tragically, anencephaly is something that is basically incompatible with life. And so affected infants will often be stillborn, or will typically only survive for a short time outside of the womb. And it is tragic, but we now know that there are some contributing factors and some prevention that can take place. So there are some genetic and environmental factors. Sometimes there's a genetic predisposition, but there's also certain environmental influences, like inadequate intake of folic acid during pregnancy, that can result in neural tube malformations. And so folic acid is, along with, which is essentially a B vitamin, is something that the body uses to guide that process. And so, but if there's a teratogen, something that's toxic that is ingested by the mother at certain stages, that can also cause this as well. So a few things to kind of be mindful of there. Now, I also want to kind of just touch a little bit on neurogenesis and the process of neurogenesis, because we have this intricate process of cell migration that takes place. So we have the birth of neurons, and neurogenesis is that process of generating neurons. It's something that is a miraculous thing that happens for the embryo, not so much as you get into later life, although it does happen in a limited extent. But it's that remarkable and complex journey that occurs during the embryonic and fetal stages of prenatal development, where you have the development of the neural tube, the neural stem cells divide and give rise to the precursor cells, and which we call neuroblasts. And as neuroblasts multiply, they differentiate then into neurons. And these specialized cells are capable of transmitting electrical signals. So each neuron possesses distinct characteristics based on its location and function within the developing brain. And this process lays the groundwork for the incredible diversity of neurons that will comprise the mature nervous system. And there's also, of course, glial cells. And so alongside this neurogenesis, another essential process is gliogenesis. And glial cells are essentially those supporting cells of the nervous system. And they originate from these neural stem cells. And so we have cells that we call astrocytes and microglia that play a role in nurturing and insulating and protecting the neurons so that they're able to function properly. There's also radial glial cells that provide supporting structure and provide some scaffolding and guiding. So, and in this process, we also have neural circuits and synapses forming. So there's a building of connectivity that begins to take place within the fetus. So as neurons reach their destination and establish connections, the synapses with other neurons, these synapses form neural circuits that form that foundational building block of brain function. And the intricate arrangement of synapses enables the transmission of information, allowing us to perceive things and interact with the world. Now, this is something that is experience dependent, and there's some refinement that takes place. So as prenatal development progresses, neural circuits undergo refinement based upon sensory experiences. So the brain's plasticity, that is its ability to adapt and reorganize, ensures that circuits are honed to optimize function based upon environmental stimuli. And then finally, we'll just take a few moments and look at some of the key prenatal neurological milestones, because that's really fascinating as well. And this is where the nervous system embarks on that wonderful journey of growth and refinement that takes place before birth. So here we have the development of the brain stem and basic reflexes. With the formation of the brain stem, we have that development in that prenatal development of the formation of the brain stem, which is that vital part of the brain that is responsible for the regulation of the body's functions, so things like heart rate and breathing and digestion, and that really kind of lays the groundwork for survival. And then we have the emergence of reflexes. And as the brainstem matures, there's different neuronal nodes that develop that are essential for reflexes. And these reflexes are innate, automatic responses to stimuli. And these are critical for protecting and sustaining the development of the organism. So, for example, we have the startle reflex or the sucking reflex, and these are very early signs of neurological activity. Then we have the emergence of the sensory nervous system, and with that, the infant begins to interact with their environment in very rudimentary ways. And along with that comes a sense of touch and taste and smell, and there is evidence that fetal organisms respond to touch as early as the first trimester, and taste and smell also play a role, and these studies suggest that fetuses can detect and respond to their favorite flavors experienced by the mother. There's also hearing, and around the second trimester, the auditory system matures, allowing the fetus to hear sounds from the external environment. And this is demonstrated by experiments that show that the fetus can recognize and respond to familiar sounds like the mother's voice or to music. And there's also some visual formation that goes on here with the visual system. Although this is less mature compared to the other systems before birth, the fetus does experience rudimentary responses to light and changes to light and light attitude and intensity. So there's also some maturation that takes place within the cerebral cortex and the higher functions. And the cerebral cortex, remember, is the outer layer of the brain that's associated with who we are. It's our advanced cognitive functions. And here, this really undergoes substantial growth and complexity during prenatal development. And so there's neural proliferation and migration that takes place. So there's a massive growth. So really, your cerebral cortex is about as many neurons as it will ever have at this prenatally. But then there's this intricate connections and synapses that take place. But throughout prenatal development and early childhood, the brain undergoes synaptic pruning, which is basically a process of refining and strengthening the connection. So if an experience is not ever encountered, then the neurons that would be associated with that experience tend to die off. And that's part of the reason why in early childhood, it's very, very important to have rich, complex experiences to give the brain as much opportunity to grasp a hold of those and to deal with them as possible. So in conclusion then, the prenatal neurological milestones really kind of go through these various processes, and we have some good sense that there's a lot going on. Now, just to kind of close out, I want to just highlight what happens, generally speaking, at each of the trimesters so that you have a good sense of what happens prenatally. So in the first trimester, we have the laying of the foundations. We have conception, obviously, in the germinal stage. And here we have, in that germinal stage, the rapid cell division that forms the blastocyst that connects and forms into and gives rise to the embryo. And we also have the formation of the neural plate. So by the end of the first trimesters, the embryonic stage, the neural plate, that flat structure that will eventually evolve into the neural tube, starts to form. And this marks that initial stage towards the development of the central nervous system. In the second trimester, we have the emerging senses and connections. We have the neural plate closing in. We have the basic structures of the brain and of the body form and differentiate. And we have the formation of the brain stem, the emergence also of basic reflexes, the startle reflex and the rooting reflex, sucking, that sort of thing. And so oftentimes you'll see pictures of ultrasounds of, in the second trimester, of a child sucking their thumb because that reflex is developed. There's also the senses of touch and of taste and smell and auditory development taking place. And then in the third trimester, we have a continuation and an elaboration of the complexity that takes place. So we're here in the third trimester. That last picture on that slide is a picture of the cerebral cortex experiencing rapid growth and complexity, getting ready, and actually is probably more complex than the environment will demand, but is ready for what is to come after birth. This is also a refinement in the sensory systems, and the time of developing real neural connections that intensifies in that third trimester, being prepared for birth. And that kind of gives us a sense of the wonderful process of prenatal development. I hope you enjoyed this lecture, and I will see you in our next lecture. You take care. Goodbye.